All right, let's get into it. We're going to talk about something absolutely fundamental. I mean, it's the story of how your body takes, say, the food you just ate, and turns it into the energy that's powering you right now. The energy for every thought, every movement, every single heartbeat. So how in the world does that happen?
I mean, it really does sound like magic, doesn't it? How does your body turn a cookie into consciousness? How does a simple little sugar molecule end up powering our most complex thoughts? Well, the answer is a process that is happening inside almost every single one of your cells right this very second, and it's called glycolysis.
Okay, so before we dive into all the nitty-gritty details, let's set the stage. We need to get what our cells are actually trying to do here. You can think of it like this. Just like our economy runs on money, your cells need a kind of, well, an energy currency, something they can spend to get stuff done.
And that brings us to our star player, glycolysis. It's basically a 10-step chemical assembly line. It takes one molecule of glucose, a simple sugar, and just starts breaking it down. And get this, the whole thing happens right in the main fluid-filled part of the cell, the cytoplasm. What's really wild is that this process is so ancient, so fundamental, that it doesn't even need oxygen to work.
So let's just zoom out for a second and look at the big picture here. You see, we start with our glucose molecule. Now the cell has to put in a little bit of energy to kick things off. That's the 2 ATP you see going in. And by the time we get to the end of the line, what do we have? We've got 2 molecules of a substance called pyruvate, we've made a small profit of ATP, and we've also managed to snag some high-energy electrons and store them in another molecule called NADH.
Okay, so here's the bottom line. The whole point of this, the goal, is to make ATP. I really want you to think of ATP as the cash of the cell. It's the universal spendable energy that pays for everything, from flexing a muscle to building new DNA. Glycolysis is just the first step in cashing in that check that is the glucose molecule.
All right, so this whole story really unfolds in 2 main acts. The first act is what biologists call the investment phase, and it's exactly what it sounds like. Just like in any business, right? You gotta spend some money to make some money. Well, our cells, they're no different. And here we go. This is it, the full 10-step roadmap for our little glucose molecule. Now, I know, it looks kind of complicated, but don't worry, we're not going to get bogged down in every single detail. We're just going to focus on the major plot points, the really important story beats, starting right at the top there with glucose.
Okay, step 1 is just, it's brilliant. As soon as that glucose molecule walks into the cell, an enzyme called hexokinase basically slaps a phosphate group onto it. Now, this costs 1 ATP molecule, but it's totally worth it because it does something absolutely critical. It changes the glucose just enough so that it can't get back out. The cell has, for all intents and purposes, locked the door behind it. It's trapped.
All right, let's jump ahead to step 3, because this is the real point of no return. There's an enzyme here. Let's just call it PFK1. It's easier. And it uses a second ATP molecule to mess with the sugar some more. And once this step happens, that's it. There's no turning back. The molecule is now officially, irreversibly on a one-way trip to the end of the glycolysis pathway.
And that's it. The investment phase is done. The cell has now spent 2 of its valuable ATP molecules. It's officially in the red, running an energy deficit. It's a gamble, right? It's taken a risk, and it's betting on a much bigger payoff down the line.
Okay, on to Act 2. This is the part we've been waiting for, the big payoff. This is where that initial investment really starts to pay dividends. Now the cell gets to harvest all that energy it's been unlocking from the glucose.
So, the absolute game changer happens right about here, at step 4 on our roadmap. That 6-carbon sugar we've been working on, an enzyme called aldolase comes in and just splits it right down the middle. Bam! Now we have 2 separate 3-carbon molecules. So why is this such a huge deal? Well, it means that every single step from this point on happens twice. We've just doubled our potential profit.
So, in the next few steps, the cell really gets down to business. For each of those 2 new 3-carbon molecules, it starts pulling off high-energy electrons and stores them in that NADH carrier. And then it starts making ATP directly. It just takes a phosphate group from the sugar and sticks it onto an ADP. And because this happens for both molecules, we get a nice little energy harvest.
So, let's tally that up. Because everything happened in duplicate after that big split, the cell cranks out a grand total of 4 ATP molecules in this payoff phase. I mean, that's a pretty great return on that initial 2 ATP investment, right?
All right, let's do the final accounting. We made 4 ATP, but remember, we had to spend 2 to get started. So that leaves us with a net profit of 2 ATP. Plus, don't forget about those two super valuable NADH molecules. They're going to be worth a ton of energy later on, as long as there's some oxygen around. So yeah, the gamble totally paid off.
So we've made it to the end of the line for glycolysis. Our original glucose molecule is now 2 molecules of pyruvate. But the story is far from over. Pyruvate is now standing at a fork in the road. It has a choice to make.
And what does that choice depend on? It's really just one thing: oxygen. If the cell has mitochondria and there's plenty of oxygen available, we call that aerobic conditions. Pyruvate's going to head down a path that will generate a massive amount of ATP. But if there's no oxygen, or maybe we're in a cell that doesn't even have mitochondria, like our red blood cells, well then it has to take the other path, the anaerobic one.
And you can see that fork in the road right here. Under those anaerobic conditions, think about when you're doing a really intense workout. Pyruvate gets turned into lactate. Now, why would it do that? It's actually really clever. This reaction recycles a key ingredient, NAD+, that's needed way back at the start of the payoff phase. If the cell didn't do this, the entire energy assembly line would just grind to a halt.
Okay, so now for what I think is the most elegant, most beautiful part of this entire story. How does the cell control all of this? I mean, it would be super inefficient to just have this pathway running at full blast 24/7. There's got to be some kind of master switch, right?
Just think about it for a second. If your cell is already loaded with ATP, it's got plenty of energy. So why on earth would it waste a perfectly good glucose molecule just to make even more? It needs a way to check its own bank account, so to speak, and turn the whole process down when it doesn't need it.
Well, the answer brings us back to our old friend PFK1. Remember that enzyme from the point of no return step? It turns out it's not just a one-trick pony. It's also the cell's main energy sensor. It's amazing. When the cell has a ton of ATP, the ATP itself literally tells PFK1 to slow down. But when energy is low, and you have a lot of ATP's leftovers, like AMP and ADP, floating around, they tell PFK1 to hit the gas, and glycolysis speeds right up. And the way it does this is just so cool. See, when ATP levels are high, an ATP molecule will actually bind to a special little regulatory spot on the PFK1 enzyme. This makes the enzyme change its shape, and that new shape is the off position. But when AMP is high, it comes in, kicks the ATP out, bonds to that same spot, and flips the enzyme back into its on shape. It's literally a simple on-off switch that's controlled by the cell's current energy balance.
I mean, this is just a stunning example of how efficient biology can be. The very thing the cell is trying to make, ATP, is also the thing that circles back and shuts down the production line, right at an early, crucial step. It's this beautiful feedback loop that makes sure the cell is always perfectly tuned to its own needs, never, ever wasting precious fuel. So this PFK1 switch isn't just a neat trick. It's a master regulator of our entire metabolism. It has to work right for us to be healthy. And that, of course, leads us to a final and really crucial question. We know that in diseases like cancer, cells develop this insane, bottomless appetite for glucose. So you have to wonder, what has gone wrong with this beautiful, elegant switch?